Welcome to the Chemistry, Biology and Math Revision Hub. This is the June 2020 Pearson Edexcel International A-Level Biology Unit 6. So I will highlight here Unit 6. Let's begin. This paper is 50 marks, so I'll go straight to question 1. Question 1 says, the photograph shows two species of potatoes, sweet potatoes and white potatoes. The tissue in the tubers contains molecules that can be used to produce new plants. The tissues of the tubers contain starch molecules, and they say explain why starch is an energy storage molecule. Now, starch is a good energy storage molecule because it's made up of so many glucose molecules linked together by glycosidic bonds, basically. So it means it's a polymer containing many glucose molecules that are linked together. So if starch was to be broken down, a lot of glucose molecules could be provided and a lot of energy can be generated. Another factor is starch is actually insoluble. So if, let's take an example, if this was a cell and starch is inside, starch will not dissolve in the solution inside the cell, and therefore the osmotic potential will not be generated, or osmotic effect will not be generated, causing water to come inside. If there was a situation where a molecule is soluble, it could dissolve, creating a highly concentrated solution inside, which can lead to water coming in by osmosis, and therefore causing the cell to swell and then burst. So in this case, so for plant cells, then it could be maybe increasing in size and becoming bigger, and therefore starch, if it was be soluble, it wouldn't be a good storage molecule. But in our case, starch is insoluble, so it will not dissolve, and then it will not lead to osmotic effect inside the plant cell. So the next part is the branched glucose molecule in amylopectin. So as I said, starch is made up of two molecules. There is one that is called amylopectin and the other is amylose. The amylopectin has branching glucose molecules that can be easily hydrolyzed in order to provide glucose once needed for respiration or as an immediate source of energy. So that is why it's a good storage molecule. The next part says describe an experiment to compare the water potential of tissues from these two types of tubers. Now, if we are to compare, we need to begin with an experiment. So we gather two tubers, tuber 1 and tuber 2, and then we have to cut pieces from each that are equal in size. The pieces you cut from tuber A and those you cut from tuber B have to be exactly the same size. They have to be the same length. They have to be the same mass if possible. So when we cut, we have to ensure that is possible. And then to compare that osmotic potential, what we have to do is we have to get a salt solution or brine solution whose concentration is accurately known. In this case, it's usually important to use 5 or more different concentrations. So I chose 0.5, 0.1, 0.5, 1.0, 1.5, and 2.0. It depends on what you want. So you'll set up an experiment with 5 or more different concentrations, and then you have to make sure the temperature is controlled. So the temperature of the solution as well as the ambient temperature has to be controlled. And then we will place the equal numbers of each. So if you put 3 pieces from here and 3 pieces from there, they have to be exactly equal numbers. You'll place them into the starch solution, or into the brine solution, and then observe the results of the experiment. We would presume that if you place them in a highly concentrated solution, then they're going to lose water, and therefore, or if you place them in a dilute solution, then they will basically lose water. They will gain water from the surrounding solution, which is dilute, into themselves. The next part I said, the cut tubers, of course, when you put them in different concentrations of the brine solution, we will periodically measure the change in mass, or if you measured length, so change in length, or if you use volume, change in volume. I chose periodically for 30 minutes, so every 30 minutes you come and check for 3 days, monitoring the change in mass or change in volume. And then we'll have to repeat the experiment at each concentration with the same types, same size, and same mass of the tubers. And then we'll measure the length, and we will use this measured length to calculate the mean. Then we'll plot a graph. If you want to represent your data, you plot a graph of change in mass against the concentration of a solution, and we will note at the point where the curve you have drawn crosses the x-axis, because that will be the point at which there is no change in mass, meaning if maybe that this concentration is going to be very unique because there will be no change in mass in mass, of course, of the tubers, and this is how you can represent your results basically. The next part says, state one abiotic and one biotic variable. Abiotic is non-living, And then biotic is basically living. So the abiotic I chose was temperature, or you could choose mass or pH, or it could be the volume at the beginning of the experiment. The biotic is to do with living, so I chose it could be same age of the potatoes extracted from the same part, or cubes cut from the same part of the tuber. The next part says, choose one of the variables you have identified in one and explain how this variable could be controlled. You describe what effect it could have on the results if it is not controlled. The variable I chose was pH, and we can control pH using a buffer or a buffer solution. And then if this is not controlled, the results will not be valid, because you have to explain, of course, this is about movement of water across the membrane. And we know across the membrane, there are channel proteins, and these channel proteins are very sensitive to change in pH. So if pH is changed, the shape of the channel proteins could be different, and that it will affect the permeability of course, permeability of the membrane, or the shapes of the proteins or the channel proteins being changed could affect whether water comes in more or it does not come in at all, and that can affect permeability of the membrane. So I will go to the next page. This is the last part of question 1. They say explain the factors that may affect the water potential of the potato cells. So here we have to know is depending on the composition of the potato inside, different potatoes or different cells within potatoes can have different ion composition or ion concentration, so that could affect. So depending on the species, the ion concentration inside the cells could be different. A different concentration of the ions can affect the water potential, and also the different compositions of the cell wall. So if in one cell wall there are many aquaporins or channel proteins for water, while in another there are fewer, it means the one that has more could have more water coming, and the other, or leaving out, and the other could not be. If it has fewer, it could allow less water to come in. So this brings us to the end of question 1. Let's go to question 2. Question 2, the photograph shows a thistle plant. So this plant grows in grasslands and is not eaten by cattle. A student investigated the effect of treating thistle plants with a solution of insecticide. Other thistle plants were treated with water. So it means we are treating with insecticide, and the other plants we're treating them with water, and comparing how they will be after the results of this experiment. So they say all thistle plants used were selected at random. This is very important. It means the experimentation is not biased, and they are from the same field, meaning there is consistency. After 56 days, the students measured the longest leaf of each thistle plant used in the investigation, and then they say state the null hypothesis. So what we are varying here is we have the concentration of the insecticide as well as water, and observing the length of the leaf. So it means the null hypothesis should be in relation to the length of the leaf in corresponding to the insecticide, using insecticide or water. So for me, my null hypothesis was there is no significant difference between the length of the leaves of thistle plants treated with insecticide and those treated with water after 56 days. So let's go to the next page. The next page gives us the information. Allow me to take away that. It's not that necessary. So the table shows the results of this investigation. So we have the results from treatment with insecticide and the results from treatment with water. And they say calculate the percentage difference between the shortest and longest leaves in those treated with insecticide. So here we can see that is the longest leaf. And the shortest leaf is, of course, this one. So we have to get the difference, which is 14.0 minus 11.1 divided by 11.1 times 100, giving us 26.1% as the percentage difference between the shortest and the longest. The next part, they say calculate the mean length of the leaves for plants treated with insecticide and the mean length of the leaves for plants treated with water. So here they say draw a suitable table to display the raw data. If they say to display the raw data, you have to display the data the way it is, and then we have to calculate the mean for each treatment. So the raw data is going to be displayed in a table. So a table should have labels as well as units. So I said the length of the leaf in centimeters. One part is for the insecticide treated and the other part is for the water treated. And then I added up everything and divided by 15 to give me my mean here, and add up everything here, divide by 15 to give you your mean here. So we have the two means, one for the insect treated leaves as well as that for the water treated ones. So this is how you represent to get 3 marks. I will go to the next part. They say plot a suitable graph to compare the results of this investigation. So you are supposed to draw a bar graph here. Let me kind of zoom so that you guys can see properly. So here we see on the y-axis we have to put the length of the leaves because that is the dependent variable you're trying to measure, and on the x-axis whatever we're varying, which is either treating with insecticide or treating with water. So the mean value is what you see here. That is the highest value when we treated with insecticide. This is the lowest value with insecticide. We draw a line through. And then for this part, that is the mean value when treated with water. That is the highest value and that is the lowest value. If I use a line to draw a line and see if the two error bars do overlap and I position it appropriately, you can see there is no way these two error bars do touch. So there is no overlapping, meaning the results of this experiment can actually be valid. Based on this data, because the error bars do not overlap. So also when you're getting the scale for the y-axis, you have to make sure it is appropriately chosen so that the graph you have drawn, or the graph basically you have drawn, fills at least a third of the page provided. The next part says the student analyzed the data with a t-test using the formula. This formula, you have seen it many times. So they've given us that x bar is the mean value of each treatment and n is the number of thistle plants in each treatment. They've given us S²1 and S2 squared, so they want us to get that. We have everything, we just have to substitute into here. So I substituted. Remember, this is the mean from the first experiment, and that is the second one. So 12.6 minus 9.8 divide by the values we have substituted in here, keeping in mind that n is going to be 15. And then when you use your calculator, the resulting answer is going to be 10.3. 10.3 is actually your calculated value when you do this t-test. Calculated value. So we have to keep this value because usually the next page they're going to give you the table of critical results and ask you to see if significance exists. So here we see the table which shows the critical values for T for the different degrees of freedom. The number of degrees of freedom is given by this formula. So we know N1 was 15 and N2 was 15. 15 minus 1 gives us 14, and this 15 minus 1 gives us 14. 14 plus 14 gives us 28. So the total degrees of freedom are going to be 28. So let's go continue down here. Total degrees of freedom will be 28. So since we have 28 degrees of freedom, it's going to follow that line. And we know in biology, we gather critical values basically at p is equal to 0.05. So I'll come here and there, and this is going to be our critical value from the table. The next part is asking us to deduce the conclusion that can be drawn from this investigation, and they want us to use the graph and the information in the table to support the answer. So based on the graph, based from the table or from the results we have obtained, the calculated value is 10.3, while the critical value is 2.048. So we see the calculated value is greater than the critical value. What we have to do here is to reject the null hypothesis. Remember, the null hypothesis stated there is no significant difference between the length of the leaves exposed to the different conditions, whether water or insecticide. If we are rejecting it, it means actually there is significant difference between the length of the leaf and of the leaves treated with insecticides and those treated with water in the thistle plants. And actually, because the error bars did not overlap, it means the results we have obtained here can be validated, or they can be valid. The next part says, explain two ways in which this investigation could be improved. We could improve this investigation by trying to control the environmental conditions. And again, remember, these plants grow in a field. And you know the soil in the field, you cannot control the conditions to monitor the amount of nitrate or the minerals or the water inside the soil. And this is going to be problematic. So if we can monitor or control environmental conditions of the plant, this could be we could make the results more better, or we can make the results more conclusive. So the conditions can affect, if not monitored, they can affect plant growth and lead to a difference in the length of the leaf. And also we have to measure the leaves before and after the experiment so that we can be able to our result should be the change in the length of the leaf, not just the size of the leaf. So if we can measure the length before and the length after, and then compare the changes in the length of the leaf, that would be a better way of carrying out this experiment. And lastly, we just used pesticide, but we did not vary the concentration of the pesticide. If you vary the concentration of the pesticide, it means the results are going to be more believable, or you can see a trend in our results, and this could be more conclusive, basically. This brings us to the end of question 2. I will move on to question 3. Question 3, the photograph shows one species of genus Kabomba, an aquatic plant. So this is the plant we're talking about. They say Kabomba grows in streams and ponds in many parts of the world. It adds oxygen to the water. A student observed that there were fewer Kabomba plants growing in the shaded part of the pond, and the student formed the hypothesis that the greater the light intensity, the faster the rate of photosynthesis in Kabomba plants. So they say plan an investigation to test this hypothesis. Remember, the greater the light intensity, so meaning changing light intensity, or light intensity is going to be the independent variable. Change it. And then the results are going to be observed by measuring the rate of photosynthesis. But we know photosynthesis is about carbon dioxide reacting with water to produce oxygen as well as glucose. So it means we can measure the volume of oxygen produced, and then within a specific time, and the volume of oxygen divided by time can tell us the rate. So it means in this case, the volume of oxygen produced can be the dependent variable. We measure that and then calculate the rate of photosynthesis. So when they say state one safety issue you would need to take into account, remember we are working with plant materials, and these plant materials could have chemicals that can cause allergic reactions. They could cut us, and they could have some microorganisms that can be pathogenic to the human. So you have to make sure you wear gloves to prevent being cut as well as infection. I will go to the next part. The next part says, describe the preliminary practical work that you might undertake to ensure your proposed method would provide meaningful data. Preliminary practical work is about what you will do to ensure that the conditions of photosynthesis are going to be appropriate. Remember, for photosynthesis, we need carbon dioxide, we need water, we need the right temperature, we need, of course, the light intensity. In this case, we need light. And then we need in this experiment, we need to measure oxygen produced. So we have to find a suitable way for measuring oxygen produced that will give us the correct results. And then we have to find a way of varying the light intensity as well as which intensities we're going to use. We have to find the appropriate temperature for photosynthesis and find a way of maintaining it constant. And then we have to find to supply adequate carbon dioxide as well as water to ensure that these two do not limit the rate of photosynthesis. And then we will be okay. So I will just go to the next part. So the next part says, devise a detailed method, including an explanation of how you would control and monitor the important variables. A key thing to any detailed method is to always begin by stating the dependent variable. So here the dependent variable is the volume of oxygen produced during photosynthesis within basically a specific time. So we will begin by carrying out 5 different experiments at 5 different light intensities. Here it depends on what you're working with. I can vary light intensity by positioning the source of light at different distances from where the experiment is being carried out, and I can carry out that experiment in the dark to ensure that the only light the experiment is exposed to is the light from the experimental lamp. So you allow the plant time to acclimatize. Again, before you change from one light intensity to the next, allow the plant to acclimatize before you expose to a different light intensity. And then measure the volume of oxygen produced during each period of exposure to the light using a syringe. A syringe is good at measuring volume of oxygen. At least the errors are going to be minimal. And then the next part, we calculate the rate of photosynthesis by dividing the volume of gas produced over time, divide by the time in which it was produced. Of course, give excess carbon dioxide to ensure that it does not limit the rate of photosynthesis. And then we will control the variables like temperature, pH, and background light. These are abiotic variables. pH can be controlled using a buffer. Temperature can be controlled using a water bath. And then background light can be controlled by you working in a dark room to prevent, basically to ensure that light is only coming from the experimental lamp. And finally, we have to repeat the experiment, exposing the plant to the same light intensity under the same conditions in order to obtain the mean values. And then that will make our experimental values basically reliable. The next part says describe how your results should be recorded, presented, analyzed in order to draw conclusions from your investigation. Of course, you have to tabulate your results. The table should have labels as well as units. And then I just use as an example, if we varying light intensity like this, I expect the rate of photosynthesis to increase, provided there is no other factor that is limiting photosynthesis. So then I will get the mean value. So basically you have to use the results, the raw data, and calculate the mean. And then you'll use the mean values to draw a table, to draw a scatter graph. This graph would be a line graph or a scatter graph. This graph should be rate of photosynthesis on the y-axis with its units and the strength of light intensity on the x-axis. Then after, we can use a suitable statistical test to be able to establish if there is a correlation or not. So I said you repeat the experiment, calculate the mean values, and then for the rate of photosynthesis by exposure, we will record the results in a table with suitable headings and units, and then plot a scatter diagram, again with labeled axes and units, and find a suitable statistical test like Spearman's, it could be Pearson correlation, in order to establish a correlation basically. Lastly, suggest 3 limitations of your proposed method. The proposed method, basically, here there are some limitations we could face. Like number 1, it could be difficult to control the abiotic variables, basically, like pH and temperature, or there could be other factors that actually could limit the rate of photosynthesis that were not put into account. Other thing is the surface area of the leaves, which could be vital in photosynthesis because they do trap the light. So how can you control the surface area or area of the leaves? Because if some leaves have a wider surface area while others have a smaller surface area, those with a wider surface area will absorb more light, leading to more photosynthesis than the others. And then we also have to find a way of measuring volume of gas. Some people measure volume of oxygen produced by counting bubbles. However, if you use that method, some bubbles are going to be bigger and other bubbles are going to be smaller. These cannot be the same, so this is going to be an erroneous way. That is why counting bubbles could be an erroneous way. Another thing is if you use a syringe, even with a syringe, it could be a little bit erroneous because some of the oxygen produced may kind of be dissolved in the water before it's collected, and that could be an erroneous method. So all these could be limiting or could be basically limitations to the proposed method. So this is the end of this paper, and thank you for being with us. Looking forward to seeing you in the next video. Thank you, and please do not forget to subscribe to our channel. Bye-bye.